Over here, you see what are the stages of meiosis. Meiosis have two stages, meiosis 1 and meiosis 2. Okay, meiosis 1 and meiosis 2. As you are able to see over here, meiosis 1 is division of cell once. Right after the meiosis 1, the new daughter cells divides once more. This is called meiosis 2. You see here, meiosis 1, two cells are produced. These two cells again divide into two more cells. They are called meiosis. This stage is called meiosis 2. Okay, at the end of the meiosis, how many cells are produced? As you are able to see here, 4 cells are produced. Okay, but as you remember in mitosis, just 2 cells were produced. Okay, now here, recall the homologous chromosomes have the same genes. Genes are responsible for the our characteristics. You remember, I think, but are not exact copy of each other. Actually, the homologous chromosomes are have the same genes for our nose, for example. But you see, we get our chromosomes, one from our father, one from our mother. Actually, shape of the nose, genes for the shape of the nose or eyes or face, we can say, are taken from each parent. But which one is shown in your actually phenotype in your body? It depends on the strength of that gene. Here you are able to see homologous chromosomes. We have two pairs of two sets of, we can say, homologous chromosomes in our body. We have how many chromosomes? 46, right? 24, actually 23 comes from our father, 23 comes from our mother. Okay, each pair is called homologous chromosomes. Okay, they are not identical, but they have the same genes. Actually, they are working for the same type of the characteristics. Okay, if you have any further questions, please don't hesitate to ask any question in the comments or in the through the Jupiter as well. Okay, here homologous chromosomes are replicated, as you see here, guys. The one in the beginning of the interphase, the homologous chromosomes, actually it is at the end of the interphase, beginning of the, before the prophase, the chromosomes, homologous chromosomes are duplicated. You see, one homologous chromosomes become double chromatid. The chromatids are produced on the one chromosome. Which chromosome? Homologous chromosomes. As you know now, homologous chromosomes, we can start to explain what are the stages one by one. Okay, before meiosis 1 begins, each chromosome is duplicated. Okay, I explained. Okay, each half is called chromatids. As you see here, these are called sister chromatids. One of them is called one chromatid, another one is called another chromatid. Totally, this one is called chromosome. This is also one chromosome. These are homologous chromosomes. One of them comes from father, one of them comes from mother in our body. Okay, how about chromatids are connected by centromere? As you remember, centromere is also present in the meiosis as well, because it's also cell, right? What are the centromere? Centromere is the location where two chromatids join together. Okay, centromere is the location where on the chromosome two chromatids are joined together. During the meiosis 1, because there are two divisions in meiosis 1, meiosis 2. During the meiosis 1, pairs of homologous chromosomes split apart into new sets. Okay, each homologous chromosomes, this one, okay, each one will go to new cell. One of them will go to right side, another one will go to left side. Okay, and it is the main difference of the meiosis, guys. In meiosis, each chromosomes are separated from on each other in the meiosis 1. okay? Not the chromatids. As you remember, chromatids are separated from each other during mitosis, not meiosis. But during the meiosis, not chromatids, but chromosomes, homologous chromosomes are separated from each other. You will see the details in the next slide. Now, prophase 1. Why prophase 1? Because there is also prophase 2, which belongs to meiosis 2. You will see in the furthermore. As you see here, in prophase 1, duplicated homologous chromosomes pair up. Duplicated homologous chromosomes comes together in at the end of the, we can say, prophase 1. As you see here, it is not mentioned, but I want to say that, you know, in the prophase, chromatins become chromosome. Okay, as you remember, chromatin is the thread-like structure which has the DNA and protein, which condenses, then becomes chromosomes. Actually, during the interphase, you know, DNA is present in our cells in the form of chromatin, not chromatid. Yeah, not chromosome, okay? Chromatid or chromosomes, both of them same. Okay. Now, here you are able to see in the prophase what's happening. Definitely, you will see in the next slide. Nuclear membrane will be disappeared. Okay, the chromosomes will be seen much more clear in the prophase 1. Now, metaphase 1, as you remember, metaphase, in the metaphase 1, the chromosomes line up on the equator of the cell. Here, that is the difference. You see here, in here, chromosomes actually comes as a pair. Homologous chromosomes line up on the equator of the cells, in the middle of the cells, in pairs. Okay, this is the difference of the metaphase 1 from the metaphase 2, we can say. Okay, from the metaphase of the mitosis, we can say. This is so simple, what just chromosomes, homologous chromosomes, line up on the equator of the cells. Okay, actually there are some mistakes over here. You see here, it is shown here that the spindle fiber attach with the two sides of the one chromosome. It is wrong actually. Here, one spindle fiber attached with the one homologous chromosome and another spindle fiber attached with another homologous chromosome. Two homologous chromosomes attached on the centromere. They are not attached with the help of the, definitely here, spindle fiber. There is no spindle fiber between them, but they have drawn like that. No problem. They draw like that. Okay, leave it. No problem. Okay, homologous chromosomes line up on the equator of the cell. That's easy, I think. Okay, leave it. Now, anaphase. It is also similar. What chromosomes actually separated from each other. Here, chromosomes are separated from each other. But in the, you know, as you remember in the mitosis, chromatids were separated from each other. Here you are able to see each homologous chromosomes are separated from on each other and goes to the opposite pole of the cell. That's also simple. Okay, pass it. Now, telophase. In telophase, what happens? The chromosomes, homologous chromosomes, are separated from on each other completely. Okay? They reach the opposite pole of the cell. Around the homologous chromosomes, nuclear envelope again appears. You see? Nuclear envelope is formed. And here, one thing more. You see, actually, in the beginning of the slide, you saw that there were 4 chromosomes in the one cell, but now you are able to see 2 chromosomes are present in the one cell. Here, this is called chromosome. This is also called chromosome. Actually, there are not 4 chromosomes here. There are 2 homologous chromosomes are here. Actually, if I ask how many chromatids are present, then you might say that there are 4 chromatids, but not actually. I'm asking the chromosomes. How many chromosomes are present? two chromosomes are present. If I ask, at the end of the which stage in meiosis the number of the chromosomes become half, you will say that at the end of the telophase, the number of the chromosomes become half in each new daughter cells. Okay, then cytokinesis occurs. Cytokinesis is the division of the cell, cytoplasm in the cell. Then after the cytokinesis, two new daughter cells are produced. So cute, right? Okay, it's not funny. Now, okay, I think you understood telophase 1 as well. The completely migration of the chromosomes are completed. Okay, and cytokinesis after that followed by